So I'd like to welcome you all here tonight. My name's Tom Sperling, and I'm a member of the Centre for Transformative Innovation. It's a great pleasure for me tonight to introduce Erold Harvey to you. I first met Erold in 2002 when I came to Swinburne as the director of IRIS, the Industrial Research Institute of Swinburne. I soon realised that Erold was going to go a long way when I, as the director, had to make an appointment to see Erold. He was so busy. Erold has a PhD in plasma physics from Monash University. He went to the Rutherford Appleton laboratories in Oxford to work as an operations manager there, and he then went to, as a senior development engineer, to ISTAT. And he came to Swinburne in 1998 as the professor of microtechnology. And it was during that time when the CRC for microtechnology was around that Erold and the CRC in Swinburne decided to set up the spin-off company miniFAB, and miniFAB has now not. I don't think Swinburne has any shares in miniFAB anymore, but it's been a very successful company developing technologies that are used around the world in both aerospace and medical applications. So it's a great pleasure for me to introduce Erold to you and get him to talk on his 40 years of microfluidic commercialisation.
Thanks, Tom. Small correction, it's not my 40 years, it's 40 years of the subject. So I would have had to start quite early if it was me. Do I need this, or can you still hear me if I'm away from the microphone? Is that all right? Okay, great. Tom's introduced miniFAB a little bit, but just to give you a brief snapshot, we've been around since 2002, which is when we were formed as a spin-out. We're headquartered here in Scoresby, down in the eastern suburbs. Around 150 people now, and we have a split between manufacturing operations, largely in polymer microfluidics and medical devices, and then the other half of the business is in contract product development. So we're a service business, and we develop other people's products, and then we manufacture those things that we develop as well. And because most of our work's in the medical device area, we are ISO 13485 and 9001 and all the regulatory sort of things that you need in order to appease the FDA. Microfluidics is a lot of fun. And this is the little example, not from us, but from the Suzuki lab at University of Tsukuba. And it's a microscope slide. It's got some channels in it. And you can see 3 liquids or 3 fluids being manipulated in there. There's the green and the red, and the other fluid is air. And you can see that by having a control system manipulate the device, you can take 3 aliquots from the liquid samples. You can reorder the aliquots, and then by removing the air from in between, you can get them to mix. So in an entirely automated way, you can do what would take a skilled technician quite a long time to do with quite a lot of variability. The other device here is a little cartoon of an integrated microfluidic cassette that we've been building for John McDivitt's lab at NYU. And it shows how we use blisters to push fluids around a circuit. And then the business end is actually a set of agarose beads. Those beads there's 42 of them, each of which do a different and fully independent immunoassay. And then the microscope is a fluorescence microscope that does a readout. So in one test, you can do 42 measurements. And it's the sort of things that we're seeing happen now in what's called LabOnAChip, where you can integrate many complex biofluidic processes into something which the machine can handle. So some examples of some of the things that we've worked on. This is a device we built and manufacture for Tier Lab. We do the consumable, which is the tip of this, and it's a device for measuring exactly 50 nanoliters of tears, and within a few seconds coming up with diagnosis for dry eye. And the important thing about that is that we hold a precision on that of better than 1%, which means that we have 100 nanometer tolerances on all the plastic molding and the components in there. Another device that we developed with Becton Dickinson, this one is used for AIDS diagnostics. This is a more recent device with Himaxis. They're a spin-out company from EPFL in Switzerland. And this is a microfluidic blood sampling device which then transfers the blood to basically a filter paper which can then be transported as a dried blood spot to the pathology lab. And GenePOC is a device which we developed with a French-Canadian company. The consumable are these little, they call them pies because they're like a segment of a circle. And they do complex DNA diagnostics, particularly for measuring drug-resistant bacteria in hospital infections. And this uses the centrifugal force of the rotor, the spinner, to basically fling the fluids around the cartridge and run the assay. So here's a little bit more detail of the tear collection device. It's an injection molded piece of plastic. The business end is this tiny bit here that you can just about see the vent hole, but you can't actually see the channel. And by touching the bottom of the tear lake, the capillary forces wick up the fluids, and then essentially this instrument is a potentiostat that does an electrical measurement and comes up with a numerical output. And this is the Becton Dickinson device. This is an entirely passive fluidic system. The drop of blood from a finger prick is put onto here, and then this lid is closed, and then we peel off this protective layer and it opens up a window. And then entirely using the capillary forces of the fluid, the blood will flow through a little container which has got the stain on it. It'll stain the CD4 cells, spread the blood very evenly over the device. We've got to make sure that we don't get two red blood cells on top of each other because we want to count all the red blood cells. And then this machine is effectively a fluorescence microscope again, which does a count of CD4 and hemoglobin, which is an important marker for AIDS. And the whole thing was designed for use in resource-constrained economies, so it's on sale in Africa. So the field at the moment, the field of microfluidics, particularly for point-of-care diagnostics, is growing quite well at the moment. It's got about an 18% compound annual growth rate. The figures are a little small at the moment, but growing nicely. So it's roughly a $1 billion market by 2021. But that's just the fluidics. The actual test, which includes the biology and all the other components, is usually about 3 times more than that. So it's looking like a market of around about 26 billion when you include other areas of point of need. This is a French consulting company that's done this, and they call point of need to include environmental monitoring, food and agriculture monitoring, as well as point of care medical diagnostics. The other thing we're seeing at the moment is quite a lot of commercial activity in terms of acquisition in the subject. So last year there was fundraisings around the world of about 600 million for companies that are going into microfluidics. 18 new systems were introduced to the market, and mergers and acquisitions very active, and I think we're going to see a lot more of this, something like about 11 billion in mergers and acquisitions, not all just because of microfluidics, but in this whole area of point-of-care diagnostics. And this is a little snapshot of some of the products that have come on the market recently over the last year or so, and that we know are coming onto the market. And you can see that there is quite a growth in the number of different products which are happening. So the point about all of that is to illustrate that the commercial activity around microfluidics is now well and truly measurable. It's taking off. But actually the start of microfluidics goes back, as I said, about 40 years ago. And it actually started in a commercial setting, not in a research setting. This, as far as I know, is the beginnings of microfluidics. So this is the ICI Winnington Research Labs in the UK, just outside Birmingham or around that area. Some researchers here in the 70s asked the question, why do we always have to build such huge chemical plants to build these new chemical entities that we're researching? Why can't we scale up continuous flow reactions? And then the scale up of the volume can be by making more devices rather than the huge risk which is involved in scaling up a volume, so a reactor getting a lot bigger. And particularly for very exothermic reactions, reactions can go out of control very quickly. Early 70s they started the work on this. It really didn't get anywhere. They didn't publish. And I heard about it because I met the guys in the late 90s and they were well and truly retired. So in a sense, this early sort of start, very promising, didn't get anywhere. It is used by BASF and some other chemical companies still. They will use continuous flow chemistry, but it really didn't take off much. Meanwhile, in the United States, in the early 90s, some entrepreneurs started to look at the idea of using a silicon wafer and some very simple microfluidics to basically measure blood chemistry. And they formed a business called ISTAT and started to develop this cartridge which you can pop into a handheld unit and measure lots of different parameters in blood chemistry. Now, ISTAT really struggled. The point about this was to try to introduce complex tests at the point of need, next to the patient. And they really couldn't get it into the workflow of standard care. Famously, they had to look for niche applications like in an ambulance or in an emergency ward or on the space station. I think that's the sort of places they could sell to, but that's not a big market. So they struggled for a long time. Around 2001, they were taken over by Abbott, and it's still on the market. It's quite successful now. If you go to a hospital and talk to somebody in pathology around point of care, they usually will say, yes, I've got an ISTAT. But if you think about that, that's nearly 30 years before it actually made sensible market penetration. So it had to be very patient. In the 90s, though, this was the sort of image which was being spread around. It's what gave rise to the name of LabOnAChip. And the idea, this is a caliper image, was that all of these white coats, the little machines and the people moving solutions from one place to another, could be integrated into a piece of glass, a piece of plastic, a piece of silicon, and then everything would happen automatically. Now, around this time, this is when we started to see hype really take off. It's a lovely image. It's a very tantalizing idea. It's very difficult to do. And we had our fair share of this hype here in Australia. So this is Ambree. I know you know it very well, Alexander. Yeah. This company had a sensing system which was exquisitely sensitive. And therein lay its problem. It was so sensitive that there are some very good technical reasons why the approach they were using for the cartridge could never work. Nonetheless, the hype was high. They spent over $70 million here in Australia on this. Listed in 2001 on the ASX. At $32 million, they were the largest Australian biotech listing at the time, and failed. It's a salutary lesson because it turned a lot of investors off the whole area of biosensors in Australia for nearly a decade. So we were victims of the hype that came from this sort of thing. But of course, technology hype's not new. And let's just have a little look retrospectively at some other hyped technologies. This is the Geiger Site in about 1900. In Basel, in Switzerland. And of course, in the late 1800s and around this period, chemistry was the big science that was going to save the world, transform everything. Now, I'm not a chemist, so there are others in this room, like Tom, who know better than I about this. Not that you're there, Tom, but it's your subject, sorry. Lots of great potential from chemistry in those days. It gave us things like dyes that we didn't have before, colours, coloured fabrics and so on, that were miraculous at the time. But a mere 14 years later, we had, mustard gas factories and people who were very fearful of what these terrible chemists were able to do in very large numbers. Of course, these days, Novartis is now the latest incarnation of the Geiger Site. They've got a very funky-looking headquarters still in Basel. But we treat chemistry as, I think, a subject, a discipline in engineering that is much more balanced and measured. It's got its good sides and its bad sides. We just treat it as part of what goes on in our worlds today. Here's another one that's suffered a lot of hype. This is a atomic power reactor. I can't put a date on this image, but it must be obviously after the Second World War. I looked at this very carefully. I have absolutely no idea how this could work. I don't know what's going on in here, but it comes through there and then makes a belt go round and round. I think this is a flywheel, and that must be the generator. So, lots of potential for atomic energy. Within a few years, 1947, the hype's well and truly underway. In Britain, famously, atomic power would be so cheap, it wouldn't be worth charging for it. It would best just to give it away. In the United States, somebody was proposing maybe an atomic-powered railroad or steam engine, which actually makes a lot of sense when you think about it. The atomic cappuccino coffee machine, if you've got one or can collect them, they're an icon, 1947, still around as collector pieces. Ford suggested in 1958 an atomic-powered car. And then, of course, in 1986, we got Chernobyl, and we were already suspicious of atomic energy, but this turned everybody off it. These days, you still see atomic used in odd places. I don't actually know why this is an atomic watch, but you can get a Casio solar atomic watch. I guess it's the brand name. This is my subject area, lasers. Cover of a 1924 magazine, The Diabolical Death Ray. Einstein had only just got the Nobel Prize 1905 for stimulated emission, so we were still far away from ever being able to get to a laser. It would take Theodore Maiman in 1960 before we got the first ruby laser, and this is what it looked like. It's a piddling amount of light that came out of it, but it was the first time that we could get stimulated emission. Only 4 years later, James Bond was going to be killed by one of these things, and it was able to do practically everything. A laser would basically solve any problem that you had. This I picked up way back in about 1984 at the equivalent of Bunnings. This is called a laser draft excluder. As far as I can tell, it's a piece of fabric with some beans in it, and the idea is you put it at the bottom of the door to stop the breeze coming through. But it's long and straight, and so obviously laser-like. Nowadays, again, much more measured. Lasers are ubiquitous. From laser pointers, kids in their bedrooms have these laser cutting machines, which can chop out shapes and stencils. Most people don't have DVD and CD players anymore. That's sort of disappearing, but certainly in laser barcode scanners and fusion research in lasers still goes on. So this kind of cycle is fairly well understood or well known in the area of technology commercialization. And One of the US consultancy firms called Gartner has quite a while ago published this as the Gartner hype cycle. And what they refer to is this hype phase followed by, this is an arbitrary scale of hype, and this is time, okay? So you get your hype phase, and then you get what's known as the trough of disillusionment. Now, in microfluidics, we had the hype here, LabOnAChip, we will revolutionize medicine. And this is symbolic of usually investors saying, well, we've spent a lot of money in this area, we haven't seen anything, where are the products? We haven't changed how doctors are behaving or working and so on. So you head down into this trough of disillusionment, and then slowly, the slope of enlightenment until you get to some productive plateau later on. So I find it quite interesting to overlay this hype cycle in microfluidics, and this, by the way, is my version of these arbitrary units, with publications on microfluidics. So this is a very unsophisticated Web of Science check on any publication with the keyword microfluidic. And you can see from a relatively low start in '98, it kicks up. It's on its way down. I think we're past the peak phase of publications in microfluidics. But it's interesting to overlay these two. And I don't understand what this correlation really means. But there is clearly a lag between the hype and the kick-up in publications. And that lag seems to be around about the 4-year mark, which also is consistent with the time it takes to write a grant, get a PhD, get somebody to actually start publishing some information. So, yeah, we got this, I think, a non-correlation between publications and this. And I haven't got the resources to do it, but I'd be very interested to see what patents look like on the same time frame, and how they plot up against a hype cycle and against publications. But this comes to an issue which is very common now, and that's for universities to be concerned about intellectual property. In 2000, I remember going to a conference on microfluidics in Singapore, and one of the speakers stood up and said this is the first subject that he knew of where the mantra that you must patent before you publish has become ubiquitous in the field. And of course, now it's everywhere. So we're 17 years on. Has that idea that you have to patent before you publish, has it helped or has it hindered the whole commercialization process? And I guess this gets into a much more complicated area around patents as only one aspect of intellectual property. And there's an awful lot of gaming that goes on in patents, and you can see that particularly in microfluidics. So patent before publish is the mantra in research. That has generated a huge number of pretty worthless patents. Particularly around 2005, the US Patent Office got swamped with far too many patents, and they decided to take a pretty light-on approach to letting patents through, and they used the courts to decide whether this was a valid patent or not. That gave rise to a behavior called patent trolling, where companies would gather together all these fairly random patents, and then anybody that was trying to commercialize in this area would get hit with a lawsuit from one of these patent trolls and say, look, here are our 300 patents, you're violating us, we're going to see you in court. And it would take small businesses all of their resources to try and fight that, and usually they'd fold over. Even if they were right or wrong, it didn't matter. And Caliper was guilty of this in a big way. And the other thing that we also see is ego patenting, and I'll show you an example of that. But just this idea of university research generating useful IP that accelerates commercialization, I just want to explore a little bit further. And to do that, I need to step back a bit and just show you what a development pathway in a very abstract way looks like from a commercial player's point of view. So this is what we do with our customers. When we start a development process, we look at what the market opportunity is and we form a document that's the product requirement specification. The color key here is that anything in red is actually part of the regulatory process. Once we've defined what the market really wants, we create a concept that meets that market. We generate some proof of principles, we do some prototyping work, and this is a bit of an iterative cycle. Once that's working okay, and we've got all our proofs of principle checked out, we can start putting it together into some integrated prototype, and we can do some preclinical trials. And again, this is a bit of an iterative loop. And then once we've got confidence of that, we can go into clinical trial phase, submit to the regulator, and we can do a product launch. And then after that, we distribute, sell the thing. Hopefully people will buy it. And the regulations require that we have a vigilance system which checks that there are no failures or mistakes or accidents out in the marketplace. And this is a very important feedback loop that goes back into the design. So that's a very quick version of the design process. Let's have a look at what a research process looks like. Again, very simplified. A researcher will start with a grant application. If successful, will recruit a postgraduate. The subject has some kind of hypothesis which leads to an experimental investigation, and then you refine the hypothesis and you basically do your research. And if something good or useful has come out, you'll capture the IP before publishing and spit out the graduate student. Now, how do these two processes intersect? Well, only at one place, really, and that is on the off chance that the IP that was captured by this process happens to fit the concepts which are required in this process. Now, the likelihood of those things coming together at exactly the right time is so infinitesimally small. It's no wonder that it's very difficult to get businesses interested in the early stage IP because they've been done completely independently. And you hear different language about this, people saying, oh, business hasn't got the absorptive capacity for our new ideas. Well, yeah, because the intersection point is so tiny, it's very hard to hit that. And then when you look at the commercialization process that's usually involved in a university, what it means is that the university tries to contribute this IP and capture some of the end value out of marketing and distribution in a way that they can feed this back into supporting the research process and run this again. Now, look how big that loop is. And once again, you can see why it's so difficult and so high risk, and how the likelihood of anything useful coming out of this is very small. So I think the whole process by which we capture intellectual property and try and commercialize needs a severe rethink, because this one, there's some success stories, but most of the time it doesn't work at all. Another way of looking at patent landscape are these maps. Are people familiar with these? Anybody seen them? Yeah. Okay, so it's a Thomson Reuters piece of software that does landscaping. You can't really read it and you can't see the dots, but blur your eyes and you get a kind of a visual representation of, in this case, it's the nanotechnology patent landscape from 2007 is one I had. Now, what you find, if companies are active in patenting, and in this time L'Oreal was one of the largest nanotech patenters, you find a very strong clustering around key areas of application. There's another strong cluster here. So these are usually indicative of business activities which are very focused around a certain product or technology that they're interested in. You can also see this random scatter of dots all over the map. those usually come from the regents of the University of California or the Massachusetts Institute of Technology. And it's because there's a non-strategic way that those patents are captured. It just sort of gets splattered all over the map. What this tends to do, and it has done in microfluidics, is that whenever anybody comes to look at the subject, they first have a check if the patent landscape looks clear, usually as a way of checking if there's freedom to operate, see all these patents there, most of which are worthless or dormant, and then conclude wrongly that everything's covered and that I shouldn't be going there. So in fact, this plethora of patents has worked at a disadvantage to the commercialization process. It's turned a lot of people off the subject rather than looking into it for solutions. I mentioned ego patenting, and we in Australia, unfortunately, have the best example of that, and this is a guy called Keir Silverbrook, which, again, is known to some people here. Yeah. Keir, as you can see, is a resident of Sydney. Very, very clever man. He's done several technology-based businesses, and his whole process was around building a very, very strong IP portfolio, a patent portfolio. He got interested in LabOnAChip devices and came up with this idea, which is a USB key on which you can do a DNA analysis. And so the USB basically provides the 5-volt supply to run everything. And through here, you can put your blood sample in. It lyses the cells, does an amplification. There's a chip in here which does a visual readout, and obviously the data then goes down the USB key. Very, very clever idea. He built a patent portfolio around this. Keir is one of the, if not at the moment, the world's most prolific patenter.
I think at one time he was producing more patents than the whole of the rest of Australia put together.
Yeah. In one day, Keir submitted 237 patents in one day. Question, why? Because he wanted the record to be the largest number of patents to be submitted in one day. The game is a very expensive game. And in another part of his business, Keir was having a big argument with an investor in a different technology company he was running. In the end, he lost that argument, and he therefore lost his access to funds. We at miniFAB were then asked whether we were interested in taking up this patent portfolio, and of course we were given like 2 weeks' notice before all the expensive bills arrive. And how on earth do you go through 350 patents in 2 weeks and work out where the value is? So a very sad example of somebody that really got carried away with the IP game. And now if you really want to build a device that measures DNA, there are lots of really good solutions there, 350 of them at least, out there ready for free that you can pick up. So patenting isn't necessarily the answer to commercialization. But there's been some other problems with the disconnect between university research and microfluidics commercialization, and that's often about manufacturing. A guy in the US, a guy called George Whitesides, came up with the suggestion that you could cast microfluidic devices out of a silicon rubber called PDMS, polydimethylsiloxane. It's basically a 2-part clear elastomer which you can mix up and cast off a mold. Very, very good for research because you don't need a lot of infrastructure to get started in microfluidics. Absolutely hopeless for production because they're unstable, they absorb everything, they have no shelf life. It's really difficult to turn them into a manufactured product. And so what we found was that the tools which are used in academia for doing microfluidic research were completely different from industrial tools. These were using PDMS casting, embossing, photo etching, hydrofluoric acid etching in glass or sandblasting. They're low cost, low volume, and they can essentially be run by unskilled students. Whereas in the industrial environment, we use injection molding or web lamination, web embossing. Inkjet printers, well, while they were around, were one of the biggest market areas for microfluidics, and you needed a semiconductor plant to make an inkjet printer. Now, that is not something which academia can work with. So you've got this huge disconnect between the two. A lot of the things that are coming out of here simply are not appropriate for commercialization.
That's actually ironic, Eric, because I think George Whiteside was the one that said the interface between academia and industry is prototyping, but if they're prototyping with the wrong stuff,
yeah, absolutely. The whole premise is the wrong way around. And it's very disappointing for us when we get a researcher come to us with a PDMS chip saying, look, I've spent blah, 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 so many millions of dollars of research developing this. I've done it all now. Could you just manufacture this for us? I know I have to change the materials. And the answer to that is, well, we've got to go all the way back to the beginning, because biology does care about the material. It's not just an irrelevance. Another issue which keeps coming up in microfluidics is about standardization. There isn't one. There's no ISO standard for a microfluidic chip, and a lot of debate goes on saying, well, is this what's slowing down commercialization? People point to things like the microelectronics industry, where we had to standardize on one process, the CMOS process, before we could really drive down the cost of computer chips. Before that, there were lots of different ways to make a computer chip. There are lots of different ways to make microfluidic chips, but it's a different problem. It isn't a problem around equipment and vendors of that equipment. It's a problem about integration and making your device fit for the market. That's what's really slowed down microfluidics, and standards don't help you solve those two problems. So the market is really the key to the whole thing. And the real question for any innovation is, if you're going to introduce it to the market, who's going to pay extra for this new technology? So the proposition in point-of-care diagnostics is that you can take a sample from the patient, and instead of sending it to the pathology lab and waiting for the report and then sending it back to the doctor before he can take an action on it, you can do all of that right next to the customer and get what they call an actionable result straight from the diagnostic device. Well, that is a major disruption to the current system, and there are so many vested interests in that supply chain, who funds which bit of the step, where the insurance companies play, where the pathology labs make their money, where the doctors make their money, that in fact it's been very difficult to break into this, even though there's a value proposition that a fast result is better for the patient. But we are seeing some products out there. So I mentioned the Abbott ISTAT. That is one of those devices which is now in use in pathology labs. The Abaxis system, which is also a blood chemistry system, that's been around since '95, also out there. We're starting also to see, for example, the GenMark system is a diagnostic that checks your DNA and your sensitivity to warfarin. So it's a molecular diagnostic. And we are slowly, and it is slow, we're starting to see these things get market penetration. But I guess the lesson about all of this is that the health system in particular is a very conservative industry. And the people who are in the health system have undergone very lengthy training sessions, courses, to get to that position. And so the embedded knowledge in those people is well and truly stuck. And to try to innovate very radically in that is always going to take time, particularly because it's highly regulated. The whole process of health needs all sorts of verification and validation. It's not built as a system that you can change very quickly. And as I mentioned, there are many vested interests in the current supply chain. So if it's going to be hard to change the current health system, where are other areas that this can move into? And there are some very interesting trends at the moment in what's called personalization of health. Now, it's got a range of different, the customers for this has got a range of different labels. The healthy paranoid is one of the ways that we refer to these customers. They're the kind of people that, you know, I feel a bit sick today. I wonder if I've got some terrible disease. I'll go out, buy a test, and do it to myself. The other very good market in this area is the weekend warrior, the sports fitness fanatic who would like to measure all sorts of parameters and then, you know, relive them or share them on social media. But the opportunity to get consumers to buy their own health products opens up a new market altogether. The current health system doesn't address that, and so you're breaking into new space. And some of these things which are on the market at the moment are frankly just toys, wearables and so on, but I've no doubts that we're going to see more and more players enter into this market where it's not the health insurance or the hospital which buys the test, it's you yourself using your discretionary funds. Another area that is pretty active is diagnostics for the developing world. There's a lot of good social good reasons for developing these diagnostics. This is an example of chikungunya outbreak, which is a mosquito-borne virus. And you can see it's largely in the developing world. But of course, the discipline for us of solving these sorts of problems generates cheaper, more reliable, more robust devices. So there's a sort of a double win out of this activity. And the other area that is very interesting is what's known as companion diagnostics. This is particularly moving fast in the area of oncology, where people are thinking about immunotherapies for treating cancer. The large pharmaceutical companies have this pipeline of drugs which has got a cliff on it, where a lot of their blockbuster drugs are coming off patent, and they don't have enough very good drugs that can maintain the revenue streams. Many, especially in oncology, many of the trials are failing at the late clinical stages. And so what the drug companies are now doing is segmenting the trial so that if you have this genetic type, I want to check whether you're responding correctly to this drug. And you've got a different genetic type. And is there some segmentation? Maybe the drug works fine on you, but not on you. That sort of segmentation enables them to rescue some of these drugs. But of course, the regulator now says, that's all well and good, but if you rely on segmentation to be able to choose who this will work for or not, you're going to have to sell the diagnostic alongside the drug, because we're only going to let you sell it if you can prove that the patient responds and is part of the right population group. So we're seeing a lot of very interesting activity driven by the pharma companies in the area of companion diagnostics. So 40-year tale of commercialization. Could we have gone faster? Could we have done it better? I don't think so, because it's not a technology problem. The technology for solving all of this existed early. It's a market problem. And there's no real point in developing these technologies if there isn't somebody out there, enough somebodies, who will buy it from you. So have we finished with hype? Well, unfortunately not. Investment disasters still happen in this field. And I guess, is anybody familiar with this person? Yeah. So some not. Is it worth recounting the story? Okay. This is Elizabeth Holmes. Elizabeth is a Silicon Valley entrepreneur She dropped out of her course at Stanford in 2003 as an undergraduate because she went into stealth mode to develop a diagnostic device which on a single finger prick of blood could measure dozens of different blood-borne parameters. She called it the Edison, and for several years was developing it on the quiet. Her father is high up in the Pentagon and introduced several of his friends to her business as investors. And so people like Henry Kissinger, 98 years old, knows nothing about medical devices, ended up on her board, and lots of Henry Kissinger's friends also tipped in some money. So she raised something like about $750 million, and she only had to give away a portion of her company for that. So she became the fastest billionaire. Her company was worth $9 billion based on that capital raise. Around the same time, Rupert Murdoch also invested. He bought $125 million worth of shares in this. But many of the established investors in Silicon Valley didn't go anywhere near this. It didn't smell right because there was no people involved in the business who were from our community, from the medtech, biotech area. Also, there was no information that had been independently validated through clinics or through university research. There was no papers, no publications. So again, it didn't smell right. So the savvy investors didn't go anywhere near this. But then the Wall Street Journal did an investigation on this, started to publish some reports that suggested that the tests which her company was doing were not actually done on the Edison at all, but were done on standard laboratory equipment. And what's more, they were diluting the samples in order to get it to work on standard equipment. So the FDA rushed in there and found that the clinical pathology process which Theranos was running actually broke 32 different rules in terms of running. So Elizabeth was banned from running a pathology lab for 2 years. That was sort of September last year. She is now being sued by all her investors. I think she's currently being sued for about $256 million. They've got $125 million in the bank. She was the pin-up girl for women in entrepreneurship. You can see her hero was Steve Jobs. She always used to wear this turtleneck jumper thing. It's going to be interesting to see how it all pans out, and probably later this year we'll have a Hollywood movie about it as well. It's already being made. A lot of people lost a huge amount of money through that.
And Walgreens, the chemist, got involved too.
Yeah, so she, in one state in the United States, Walgreens signed up with her, and you could go and get your finger prick of blood taken at Walgreens and wait for the test result. It turns out in the lawsuits that Walgreens didn't do any due diligence either. They just said, no, we like the... And she's a very convincing speaker. TED Talks, lots of YouTube talks. You can go and be entertained by her hype. So what's the lesson for us in all of this? There are still lots of topics which are very heavy in hype at the moment. And here's a list of some which you'd recognize. Well, I think it's inevitable. It's always been there. It'll still be there. Some of my investor friends in Silicon Valley love pumping up hype. I've realized that you can make money, a lot of money, by making small amounts of money from lots of people, or by getting a bucket load of money from a small number of people. And the hype is typified by that big bucket of money from a small number of people. So there are some vested interests who love hype and manage to time it just right. We're seeing this on the Internet of Things. Time it just right to swoop up a lot of money. So you can play that game, but just know where you are in the hype cycle. I think there is definitely a need for a different engagement model between universities and academia. Universities are very bad developers of useful intellectual property because the disconnect is quite vast, but very important validators of other technology, just as that Theranos story. The fact that there was no independent validation, no published articles, no clinical reviews, made sophisticated investors not touch that. So I think there is a definite role, and it's quite easy to understand in the clinical medical area. I think in the engineering field, we've also got some other opportunities where we can get a different engagement model. Patents are commercialization tools. They're not KPIs. If they are, they're very, very expensive ways of filling your CV. Disruptive technologies are very attractive to investors, but whether you can make any money out of it is always constrained by the economic ecosystem. And if you've got big systems like health, energy, they're very conservative areas. So you have to be prepared for a very long haul. And I guess the other problem with all of this is you can just be too early. I think ICI was too early. It's a point which, you know, many people will nod at. And I gave a similar point at a different talk in another forum, and somebody asked me afterwards, how do you know if you're too early? And I really don't know. If you've got any ideas, that'd be really good to hear them. So I'm going to end up there, and hopefully some points for conversation. Thanks very much indeed.
Are there any questions for Erold? Tony?
Erold, I was struck by the sophisticated topography of some of your slides, and particularly one about the intersection between the vertical and the horizontal. An older paradigm that you had not addressed was simply the fact that it takes 20 to 25 years between the discovery and when you get a laser-driven product in your household.
Yeah.
That was just an example of it.
Yeah.
What would be your comment on?
So I've heard that. Well, yeah, I remember somebody saying 13 years from discovery to something that is marketable, and 30 years before it has GDP impact. And I've always remembered that and tried to test that against different technologies. Has it got shorter or not? There's a perception that we're seeing faster innovation. People often say that, you know, the progress is faster. I don't think it is faster. I think it's still taking that amount of time, but more got started a while ago, and so we're seeing more of them coming to that 30-year time mark at once. So there's a perception that it's faster.
So
but
the horizontal axis of your hype cycle is really time.
Yes.
And can you quantify that?
Well, I think the 13-30 year is about right. If you're an electrical engineer and you know control theory, that thing looks like a critically damped oscillation. And so a very interesting comparison into business schools will be, what is the equivalent of the PID loop, which electrical engineers use to critically damp an oscillation in an electrical system? Because in the commercial world, there must be an equivalent driver, a set of brakes, and a dampener. And I think if we could understand better that, or maybe Beth already knows.
I just have another question. Could you say more about your idea about new engagement models between industry and academia?
Yeah, I got a more complicated diagram, which I haven't put into this slide. This one. This is the obvious intersection point. But if I go back to, there are commercial service providers for every step of this way. You can do it all yourself if you want. You can find different companies who can help you at every point along that way. And some of them are very large companies, some of them are just consultancies. So an obvious opportunity is for first of all, there to be a link between here and here. Why should this grant application be independent of a process that's already started? That's an obvious one, yeah. But particularly in these trial phases, the experiments which are going on in universities could easily be the home of some of these trials. And in the clinical trials, that's taken for granted. You always do your clinical trials in a hospital or a place like that. Why can't we find an engineering equivalent where some of the grunt work of getting the ideas nutted out, the performance understood, could be done by organisations that have the infrastructure and the facilities for doing that sort of thing. So I think it's a case of looking along this pipeline, and where in this can you form some other arrows that might join things up.
I think the challenge, Earl, is if the universities look transactionally at that step, they miss those opportunities.
Absolutely.
Along the line.
Yeah.
And wouldn't it be important, Errol, also to have a link from your left-hand market opportunities thing feeding into that vertical column?
Absolutely. Yeah.
That's probably the most important missing arrow.
Yeah.
Do you think, Errol, that the CRC was a new model of engagement in a way? What do you say about that as a model? Because that combined a lot of those.
It did, yeah. It allowed a lot of interaction that didn't happen before. So shared language, common language, and so on. I think the thing that flawed a lot of CRCs is still this thing, IP. There's a focus on patents and on this commercialization loop as somehow feeding back to sustain the whole system. And I think too many people get too clever about this instead of building the relationship that gets an ongoing interaction.
Michael and I have written a bit about the 30-day contact lens, which came out of the CRC for high research technology, which was quite, I mean, it only took 3 or 4 years from the idea to it being on the market, 6 years, I think.
Yeah.
And that was because all of those things were thought out right at the beginning.
Yeah. I think you're right. If you stop thinking of the transaction, that there's an output from this that has to be valuable to this, and instead build the loops, you're much more likely to get a useful outcome to both parties. Yeah.
Errol, just following on from that, so probably the critical mistake is that we have treated IP as a KRA, sort of by incorporating it into the ERA and getting a quality.
Absolutely, yeah.
We took that out, in fact, almost said if you're holding a portfolio of IP that you're not using, you'll deduct.
Yeah.
We might see that stop.
So when we started miniFAB, we put up a business model that said we're going to be a service model. We're going to do this development process. We don't have patents. We don't have products. We're going to go out and sell our services to others. We put that up as a suggestion to some state government funding, and the reviewers killed it. They said, this is crazy, no IP, no products, and no customers in Australia. How on earth could this ever work? And I think, again, too much emphasis on IP leads to products, leads to revenue, not realizing that IP also means know-how, that there's a tremendous amount of skills and processes that you can build in a system. The university process has a lot of processes already in it. That's IP. It's not patentable. So we've got too many patent lawyers. I heard a frightening number last week. I haven't been able to check it's true, but apparently this year we're going to graduate 15,000 more lawyers than we need. 15,000. That's a lot of people going to be trying to make this more complicated than it needs to be.
And Swinburne's making its contribution. so any...
miniFAB, I guess, making some great products into How many other companies, I guess, are in this field, and what sets miniFAB apart?
They're not a large number of companies, so our direct competitors is probably about 3 or 4.
Internationally?
They're all international. 2 in Germany, 1 in the Netherlands. US is surprisingly weak in this area. What sets us apart is the process integration that we've done. A lot of the people who are in this field have set up their business around a specific process. A good example is Microfluidics. They had a process for laminating films to create microfluidics, and they had some designs over valves. It was their IP, their patents, and if you wanted to engage them, then you had to get a royalty or pay a license for the use of that. We started with a clean piece of paper and said, look, there's a lot of ways to solve this problem. You own the IP if you pay for it. And that's turned out to be a real winning model in terms of how our customers have responded. And then we've responded by integrating all the process steps that we need. So, for example, we're putting a lot of investment into biology at the moment. We've got to build up a team that understands molecular and immunochemistry and can speak engineering. So if anybody knows of a microbiologist who fixes her motorbike on the weekend, please send them to us, because we can't find microbiologists that think like engineers. But it's that sort of integration which sets us apart from others.
Erold, really enjoyed your talk. You touched on a number of points in my history. I'd really be keen on your comments about the difference between patents and industrial secrets. I mean,
yeah.
You say you own the IP, but do you still maintain industrial secrets?
Yeah, absolutely. Yes, that's a major part of what we do. And so we do that like any consultancy. We grow our know-how by solving many, many problems for lots of different people. We make very sure that there is no leakage of one person's key ideas to another company, but we build our skill sets by solving all these problems and then resell it. And that's what most consultancies do, or any other service provider. So yeah, that's the real heart of what we do. And also when we manufacture, a lot of the know-how to hold those tolerances, we wouldn't publish. You invest in the manufacturing people and the automation equipment to meet those exacting tolerances, and you hold that pretty close.
On a completely different subject, I was interested to see most of your examples concerned blood, but there was one about tears. Now, there's other fluids, such as breath analysis, and dogs can detect cancer, etc.
Yeah.
Urine, of course.
Yeah.
Where do they stand in the hype cycle?
So,
dogs get ulcers and that sort of thing. There's a lot of hype about that, but is there anything on the...
So when you have to get your product onto the market, the fastest way to get it through the regulatory approval is a process called a 510K. And what you do with that is that you show to the FDA that your diagnostic is equivalent to the gold standard in the pathology lab. Right? If there isn't a gold standard in the pathology lab, it's a very expensive process to build the clinical data that validates that. So that's why so many people go for blood, because a venous draw is already the standard way to care. And so your fastest route to market is through a blood sample. Tears was a problem. There isn't... You could probably do the measurement using an HPLC or a GCMS or something like that, but you can't extract enough tears from the patient in a meaningful way. So our 510K submission on that was quite tricky, because it's a completely radical way of doing the measurement, and in fact the default measurement was very faulty. Breath, there haven't been the products out there in the market yet, so you've got a longer pathway. Urine is tricky for microfluidics because there's just too much of it. Yeah.
Martin.
Thank you for your talk, fantastic talk. Where are your customers mostly located, and how difficult has it been to be based in Australia? And in the current climate of acquisitions, how tempted or what are the forces that might press you to relocate in another environment, and where and why?
So more than 85% of our business is North America and Europe. Very, very tiny amount here, and we haven't really tackled Asia at all. So our customers are all somewhere else. There's often a question that comes up with them: why should we contract somebody that's on the other side of the planet to do this work? And that's a difficult one when you're young, because you don't have a track record. Later on, track record says, well, it's obvious because we're the best. And then we can validate that. We can prove that we've got the record for that. But you're right, we do have to work at it. Because we're in a different time zone, a different place, we have trained our scientists, engineers, manufacturers, our entire staff very carefully on how to interact with people through video conferencing, through emails and so on. We take a lot of care around communication. And when we do customer feedback studies, we find that's always the first thing they comment on, that miniFAB is easy to work with. And really, it doesn't matter if your partner is round the corner. If they're never there, if you only visit them once a month, they may as well be on another continent. So we don't take it lightly. We work hard at it, but the results turn out to be well. And these days, communication's pretty easy. You know, through teleconferencing, Skyping, and so on. It really doesn't matter where you are.
Philip, I'm a member of the Burundara Senior Entrepreneurs. We're in our 60s. Now, we won't see the results of these for 35 years or something. So is that a nonsense to ask older people to be inventors, or is it really a young person's job?
I think we got to be careful about the definition of IP. And I've spent a lot of time talking about patents, but of course there are the other three forms of IP. And know-how has already been mentioned. It's one of the most important. I've noticed that know-how is usually associated with grey hair or no hair. And it is something that takes time to learn. If people are savvy about how they're exchanging the important information, you're going to get it from wherever you can. And I don't see an issue with age, or I think it's irrelevant.
Just following on from Michael's question about being located here far from your customer markets, is Melbourne a good environment for you to embed yourself in? I mean, do you get the necessary support from peer organisations, suppliers, you know, just that whole network of industries that enable you to be efficient and on the frontier?
There is adequate support. I think, like all businesses, you should work at growing the relationships with researchers, with CSIRO, with universities, and so on. It doesn't just drop out the air. Really, there are only maybe 2 or 3 other places in the world where it would be different, and that would be Silicon Valley, Boston, or maybe the Cambridge area. And so the rest of the world is all going to be like us at Melbourne. So I think we are extremely well set up. We've pretty well any problem that comes up, I can usually find somebody in Melbourne, a clinician or a researcher or somebody that at least knows something about the area or can point me in a direction to somewhere I should know. So another thing we've noticed is that we're far away from our customers, but if I can get a prospective customer on a plane and get them to land in Melbourne, we nearly always get the job. And that's because they're just so blown away by everything that's here and how connected it is. So, yeah.
Yes, I endorse that.
Any other questions? So I'll ask Beth to propose a vote of thanks to Erold.
Erold, it's a great privilege. I think
Thank you.
everyone would agree to have you come out and speak to us. It's, I find it very heartening and positive to hear such success stories here, right here in Melbourne. So thank you very much.
Thanks very much indeed.
We look forward to you coming back.
Thanks very much. Pleasure.